Okay, well, thanks for coming in the rain. All right, so today is really the kind of the second to last class, and it's the last class that will have a lot of new material in it, and it's also going to be a little bit different from the other classes we've had before, in that before it's kind of been I've been talking a lot about the biological side of chromosomes, so focusing on chromosome biology and behavior and evolution and genetics and things like that. But this class is going to be more kind of focused on the technical side of how chromosomes are studied. And in particular, I'm going to talk about, it's going to be mostly focused on the microscopy aspects. And I hope some of it, I hope it's not too basic. I realize that I've shown a lot of pictures of chromosomes and chromosome binding proteins, and just kind of shown without comment. In red we have this one chromosome protein, and in green we have another one, but not really telling you how the principles behind how all that is detected. For some of you it may be really familiar already, but hopefully even if you understand the basic microscopy techniques, there should be some new stuff for everybody. Okay, and then I will finish with a non-microscopy but sequence-based method for studying chromosome structure. Okay. So anyway, back to microscopy. So microscopes have always really been at the center of research on chromosomes ever since people took the first images of chromosomes like this and wondered what they were all about. And it just reminds me that for some of the next pictures I'm going to show you, you need to wear
Thank
these. And you can keep these as yours. I have hundreds of them now. So, I have a few years supply of this. So, that's all we need to. So, yeah, 100, 150 years ago, people were taking images like this. This isn't gonna, of course, this is a two-dimensional image that's actually been squashed pretty flat, so there's no extra information there. But nowadays, pictures we have are more like this, and I'm gonna get back and see if it works from that position. It works on my monitor, but yeah, okay, I think that's pretty good. So, yeah. So you can see the kind of images we can take now are a little bit more informative. So it's not just one single plane, but this is, you can see this has a lot of three-dimensional volume. And maybe I should turn off all the lights. Yeah, so this is at the same cell stage as this one, but of course you can tell that there's a lot of problems here. And this, so this is, can anyone tell me what stage this is of the mitotic stages? We talked about the different stages of mitosis. It's the same as this, and this one might trigger your memory a little bit more. Remember what stage this is? Yeah, so this is an anaphase. So it should be, all the chromosomes should be separate, but you can see there's actually one that's still stuck. And in fact, many of these chromosomes haven't even grabbed onto the spindle yet because there's a lot of chromosome fragments that are missing their centromere. So anyway, the point isn't really to focus on the biology, but to explain to you how we can take images like this. Early microscopy is always mostly just using all sorts of white light. I'll tell you guys when there's a 3D image. There's going to be a few more coming up later. But if you use this for not 3D images, you might get a headache. So let's not use it. Yeah. until maybe the 1960s, I guess, most microscopy images were just white light microscopy. And people put a lot of effort into developing different kinds of white light microscopy, such as phase contrast and differential interference contrast, that exploited the different light bending properties of different parts of the cell versus the cytoplasm versus the nucleus. And we could, lots of very interesting things could be observed. So this white light microscopy is really quite powerful. You can see things like trafficking of intracellular vacuoles, and some things inside some organisms are colored to begin with, so you can just see them by their color. And then if you use polarization microscopy, you can exploit the fact that light has this property of being polarized. And so if you have anything in the cell which has a lot of repeating units, like microtubules, for instance, a microtubule spindle is very, can change the polarization of light in different ways. So microtubule spindles can show up if you have a microscope that's sensitive to polarization. So these all use different methods to generate contrast, and contrast is simply in your image at the end, you have to have something that's dark and something that's lighter. If everything is the same shade of gray, of course, you can't see anything in your image. So microscopy is all about generating contrast in your images. But these kinds of images are really kind of, although they're cool, they're not, they don't have the kind of information that we want out of modern molecular biology studying of the cell. Because the cell that we now know has, of course it has these organelles and structures in it, but those structures are made of very particular protein molecules. In fact, there's millions of different kinds of these molecules. We've talked about a very small fraction of them. We've talked about the names of different proteins. But we want to see actual proteins, and not only just proteins, but sometimes we want to see various kinds of proteins, or just modified proteins. So how can we use microscopy to look at individual molecules? So this is just an example, like if every different colored squiggle is a different protein, but they all have the same refractive index and they all go to the same part of the cell, how can we focus on, say, if we want to see just where these green protein, this particular protein is, how can we see where that one protein is, and how can we track that inside a cell? Because of course they're not really, proteins are not colored, at least not most of them. So how can we look at a single protein in this kind of messed up collection of millions of different proteins? Okay, there's not millions of different proteins, there's just many tens of thousands of different proteins. But we can, modern microscopy has this really nice tool where we can take advantage of a system that did exactly, evolve to do exactly that, to pick out one very specific protein among a huge heterogeneous population of other biomolecules. And that's, of course, the immune system. So our antibodies that are produced in vertebrates have this very unique, have this very interesting variable region on part of their protein. So antibodies are made of two kinds of protein chains, one heavy chain, which is big, and one light chain, which is small. And where these heavy and light chains meet, there is this, all antibodies have a different sort of pocket, and each pocket can bind to a different sort of antigen. So in principle, every antibody could recognize a different kind of biomolecule. And we need to be able to recognize every possible protein that nature can make, because you never know what new sort of protein a virus or bacteria is going to come up with. So the way we can fight off attacks and invasions from novel viruses and bacteria, whose proteins we've never seen before, is that our antibodies already have within them this site that can bind to those proteins. And we can make new antibodies, of course, in response to proteins that have been injected into us as part of some sort of attack by making some immune response. So if we take one of these poor animals and inject a large amount of this protein that we're interested in seeing, then we can force that animal to make a lot of antibodies. We don't just inject that one protein, you also have to inject some other, it's called adjuvant, some other substances that create this immune response in the animal and force the animal to think that they're under attack and stimulates their immune system to make a large amount of a single antibody. So right, back to this kind of squiggly green protein. If we injected this protein into a rabbit, along with a bunch of dead bacteria and other things that will make the rabbit make this immune response, then they will make an antibody that has a pocket that can actually recognize that green protein. And they make a lot of that, so if you withdraw blood from the rabbit, you'll be able to purify a lot of these antibodies. Of course, they don't recognize the entire protein, they usually just recognize one part of that protein. So an entire protein is maybe the same size as an antibody, but we can just inject one little part of the peptide, one little part of that protein sequence, and the rabbit will make an antibody against that little part. And that's really handy, in fact, because that means we don't need to actually synthesize or purify the protein we're interested in. We don't even have to be able to have the protein at all. We can just look in the genome sequence and find the protein sequence we want, and then chemically synthesize a very small bit of that protein. Maybe 10 or 11 amino acids is enough to make an antibody. Okay, and then so this antibody, what you do is you add this antibody to this collection of molecules, and it will sit down where the protein of interest is sitting there. So there can be millions of other molecules inside the cell, but our antibodies will only go where there's this molecule. But the antibody itself, of course, is not fluorescent. And this is an important technical distinction, but usually the antibody that we get out of this rabbit is not going to be fluorescent. So far, we've just put this antibody on this in the cell, but we still can't see it because these antibodies aren't fluorescent. So how do we see this antibody? The main technique that we use is we make another antibody that recognizes the first antibody. So what happens is, the antibody that I showed you a diagram of is actually, of course, it's a protein itself, and different animals have different sequences to their to this part of their antibody, to this constant region. So if we flip this upside down and focus on the constant region, this first antibody that came from a rabbit, it has some particular shape as well in the constant region. And so we can take just that constant region and inject that into another animal, right, and make another antibody in this different animal. So usually for this second animal, we pick a larger animal that we can make a lot more of. A goat or a donkey is a very common choice there because we will be able to make a lot more of this antibody. And so what this new antibody does is it will recognize all antibodies from a mouse. So that's really advantageous because then, let's say you have this first antibody that's not fluorescent sitting in your cell. Then you take this second antibody that is fluorescent and then add it to the first one. Okay, and now we have this fluorescent spot that marks the protein that we're interested in. And the reason we do it that way, I mean, there's many reasons why it's advantageous to do it this way. First of all, in order to make this antibody fluorescent, you have to do this chemical reaction to put this fluorescent dye onto the antibody. And that chemical reaction does not have 100% yield. In fact, you're going to lose a lot of this antibody that you started with. So if you put in 1,000 antibody molecules and try to put this fluorescent dye on it, maybe only 1 in 20 antibody molecules will actually successfully be labeled. And also you will lose a lot of the antibody molecules just in the process of making it. So you'll end up with a lot less than you started with. So this antibody that you made in a rabbit or a mouse, you don't have very much of it. You maybe only have a couple of milliliters of this antibody. So you don't want to waste it in this chemical reaction by putting this fluorophore, this fluorescent dye on it. But if you got this antibody, this second one, from a donkey or a goat, you probably have a lot of it. You probably liters and liters of donkey blood with this antibody in it. So you can afford to use it in an inefficient reaction of chemical coupling. The second reason why it's good to use this primary-secondary system is that each time you make this fluorescent antibody, you have to choose which color you want. So in this case, it's green color, but you might also want to be able to visualize this protein in a red color or a blue color or some other color. And if you put the fluorescent molecule on this primary antibody, you have to use up all of your primary antibody on these different colors. It's much easier to just call up a company and order different colored donkey anti-mouse antibodies, so you can have all the colors you want. So the actual staining process is kind of like this. So let's say this is the cell sitting down on your cover slip with all sorts of different molecules in it. The first antibody, the primary antibody, is added in a huge excess, right? So your antibodies really outnumber all the other proteins there. And then when you wash away all the antibodies, the only ones that stay behind are the ones that are bound to the protein you're interested in. And then you just repeat that with the fluorescent antibodies. And when you wash those away, then the only ones that will stay behind, hopefully, are the ones where you had your protein of interest. So with that process, we can have this cell sitting down on our slide and visualize with the fluorescent dye only the things we're interested in. Okay, so this is one, probably the most important technique, and I talked all about this technique, but I didn't say the name of this technique. It's called immunofluorescence. So the immuno part is because we're using antibodies, and the fluorescence part is because it's a fluorescent dye. So that's the general idea behind immunofluorescence, and I've said all along that we're detecting proteins with this, and it's the most common use is to detect proteins, but antibodies don't necessarily need to recognize just a protein. You can make anti-DNA antibodies as well. You can make anti-lipid antibodies, so antibodies can recognize many different kinds of molecules. So I've already said that specificity. In a previous class, I mentioned this dye DAPI many times. So in addition to antibodies, we can get fairly specific staining by using dyes such as DAPI that bind specifically to one type of molecule. Although dyes that are really good are kind of rare because, so DAPI is kind of a rare exception in that it only binds to double-stranded DNA. Most other dyes are not quite so discriminating. So it's almost impossible to find, for instance, a dye that binds to one particular protein. The structure of DNA is kind of unique in that manner, in that it admits many different dye molecules that specifically recognize it. And then the last source of specificity I've also talked about many times is this technique called fluorescence in situ hybridization. It's kind of like immunofluorescence, except instead of using fluorescent antibody, you use a small DNA molecule that is complementary to your chromosome sequence. So if you know the entire genome of your organism, then you can design this FISH probe, it's called, this fluorescent in situ hybridization probe, to light up any region on the chromosome that you want to. And so because this probe is here used against meiotic chromosomes, we can see that, of course, there's 2 spots on each chromosome because they're paired together. So this is one way we can assess whether pairing has succeeded or not. Okay, so this is pretty good. We can get very specific and see any protein we want. We can see DNA. We can see particular sequences of DNA. But the other thing is I've been showing you two colors all this time. And I should explain a little bit about how the microscope actually lets us see multiple colors. So that will require just a little bit of review of what light is. So of course, visible light is just a small part of the entire electromagnetic spectrum. Light of different wavelengths has different energies. And you can kind of, maybe if you remember this picture, you can remember that blue light, so at the short wavelength end of the spectrum, has the highest energy. So this very energetic blue photon is hopping a lot, whereas the red photon with a much more loping gait has much less energy. So a single photon, a single particle of light, will have different energies depending on its wavelength. And it's good to kind of keep in mind what these wavelengths actually are. So, so red light is about 700 nanometers, and purple light is around 400 nanometers. And only in between those boundaries, that's all the light we can actually see with our eyes. It turns out that that's also we also use these wavelengths more or less between here and here as fluorescent dyes in microscopy. But we're not usually using our eyes to detect light in fluorescence microscopy. So you might ask, why don't we use other parts of the electromagnetic spectrum for microscopy dyes? We could be using ultraviolet or X-rays or microwaves for detecting fluorescence. Why do we choose to use the same visible spectrum for fluorescence microscopy that happens to be the same spectrum as what our eyes can see? And there's kind of a simple answer for why we use the same spectrum. And it's, can anybody think of it? Like why do we use red and green and blue fluorescence dyes in microscopy and not things from other parts of the? Well, it's kind of an unfair question because there just really aren't any other dyes. In fact, the whole chemistry of the dye requires that light of this part of the spectrum interacts with those sorts of chemicals. So I guess what I'm trying to say is that this part of the electromagnetic spectrum is not just an arbitrary slice. It's actually a rather special slice in that it's the group of wavelengths, or it's a group of photons that are able to interact with electron orbitals and cause changes in electron orbitals. So the phenomenon of fluorescence really only occurs in this wavelength. Infrared and ultraviolet do not induce fluorescence in molecules. They induce other sorts of chemical changes. So UV and below, they can break chemical bonds, whereas in infrared and microwaves, they can cause molecules to vibrate, but they can't specifically excite electrons, giving them more energy and letting them release it as fluorescence. So the fact that we use visible light in microscopy, like the dyes we use in microscopes are very similar in structure to the dyes in our eyes that react to light. Not really a clear explanation of that, but I hope you get what I'm trying to say. So for instance, so a dye like fluorescein will accept light of kind of a blue. I've drawn it as purple here, but it will absorb light of a small wavelength and a high energy. And when it absorbs this light, the electron gets excited, but then it doesn't immediately, this electron doesn't release exactly the same amount of energy that was put into it. There's always a little bit of energy loss. So the electron always loses a bit of energy before it jumps back down to a low energy state. And that's why in fluorescence, you always have this shift in wavelength. So the wavelength of light you use to excite the molecule will always be different and always be a shorter wavelength than the wavelength of light that is finally emitted from a molecule. And that's why, so fluorescein, for instance, we shine blue light on fluorescein molecules, but green light comes out of it because blue is a shorter wavelength than green. So this is this aspect of fluorescence is what we can use. We can take advantage of this to do what's called, I guess, spectral separation. And we can look at many different kinds of fluorescent molecules in the same sample as long as the fluorescence profile is far enough away that we can block all the light of one color. So if we want to see something that's green, we can shine blue light on that thing that's green. So there's really bright blue light covering this entire slide. And some of this blue light will actually reflect all the way back to our eyes or to the camera unless we block it. So we have to block it with this thing called an emission filter. And we also block some of this light with this mirror. So this mirror will only allow green light to come back. So even though the blue light that we use to excite this cell is maybe thousands of times brighter than the fluorescence coming back, we're blocking it all with this mirror and this filter, and so only green light comes back to reach us. Okay, that's so this is why we see only green molecules up on that camera detector. Then if there's, for instance, red molecules in the same specimen, then we can use green light instead to excite those red molecules. So green light comes down, again there's really bright green light down here, and red light is coming back. Of course there's a lot more green light coming back, but we block that with this filter. And then all we have to do is we take separate pictures. So we take a picture of all the green molecules, then we take a picture of all the red molecules, and then we overlay them together into one, 2 or 3 or 4 color image. The main reason we take different pictures of each wavelength, well, first of all, so we can know what we're looking at by which filter we were using. And also, although there are color cameras that you can use with microscopy, they're never as sensitive as black and white grayscale cameras. So we always use grayscale cameras that just pick up all intensity and the camera in itself doesn't really care what wavelength is coming into it. So all the raw images that we take on our microscope are just black and white. And so when we put them together into images like this, we can kind of choose whatever color we want to make it look good. Okay, so that's how we deal with. We can have different fluorophores with different wavelengths and see many things in the same cell. However, these filters are not infinitely good at throwing away one color of light. So this visible light has a spectrum. It's not, of course, it doesn't come in just discrete colors of red and green and blue. The real physical attribute is the wavelength, and that varies continuously from 400 to 700 nanometers. So you might think if we had filters that were fine enough, we could have thousands of different antibodies, each with a slightly different molecule attached to it, and we should be able to see thousands of different molecules in the cell. The problem is that filters, as we have them today, they cannot really discriminate at the single nanometer level. So if we have a green filter, it's going to let through light that's about maybe 30 nanometers or so. So we can't pack an infinite number of dye molecules with infinite number of different wavelengths. In practice, 5 or 6 is the maximum that you would ever be able to visualize at the same time. And for most purposes, 2 or 3 is enough. Okay, so we have different wavelengths, and the other aspect of modern microscopy that is really important for seeing 3D things like a nucleus is being able to deal with light from above and below the plane of the cell that's in focus. So microscopes have to be able to deal and to treat the problem of out of focus light. So what I'm showing here is a picture, if you can see this, of a single intensely fluorescent latex bead that's emitting light of one single wavelength. And it's in focus here, so it looks like it's just really one single spot. And if everything were like this, it would be easy. But if we go above and below, if we go in and out of focus, so if we raise and lower the microscope stage, so we're going through focus, we can see, let's see if I can, yeah. We can see at each focal plane it's getting sharper and sharper, but if we had another bead in this focal plane, we would have all this out of focus light to deal with, and it interferes with seeing the nice in focus light. Okay, and if we take this out of focus stack and turn it on its side, it looks like this. So light from a single bead is actually spreading in all directions, and the microscope is always picking it up. Focusing the microscope doesn't mean that you're getting rid of this light. It's coming through just the same. It's just not as intense. So what are some ways of dealing with this? One really useful way of dealing with it is to use what's called a confocal microscope, where we actually physically block the out of focus light with this pinhole right here. So we actually make a hole in a piece of metal that is very small, and so only the light that's in focus will be emitted through that hole. And light that's out of focus, so for instance from here, this light that's deeper within the sample spreads out a little differently, is reflected by this mirror a little differently, and it hits this aperture instead of passing through. So only this light that is actually in focus makes it through this and into our detector. So using a physical mask to block out of focus light is one really common approach. However, it throws away 90+% of the light of your sample that you're looking at. And so it's a little bit of a wasteful method, because you're actually throwing a lot of photons on this sample and then throwing them away because they're out of focus. So there's another method of dealing with out of focus light that, in my opinion, it's a little better, but it can't reach as deeply as confocal microscopy can. So there's a bit of a trade-off here. But the method that we're using is called deconvolution microscopy. And instead of throwing away the light, you actually calculate where the out of focus light should go. So instead of rejecting this out of focus light, it's just reassigned to the correct focal plane. Just to make it kind of brief, the way we do that is with this image that I showed you before. We actually, on purpose, we take an image of a bead going in and out of focus and use that as the standard of what the microscope does to light. So if we measure this very carefully, then we know exactly how our microscope blurs a single point of light. And every picture we take with the microscope is just the sum of many individual points of light. And so if we know how that light is blurred, then we should be able to, so if we know what it looks like at every focal plane, we should be able to put it back to where it came from. And maybe I'm not going to go into the details of how it works. We can talk a little bit about it. I'll save it for the end if we have time. But anyway, it takes a blurry image like this, where there's a lot of out of focus things. So these are just mammalian cell nuclei, and makes it much sharper by removing out of focus information. And here we can see a little bit better the effect of this. So the out of focus, so it's actually a projection of the entire three-dimensional stack, but without throwing away or without reassigning the out of focus light. And in a three-dimensional movie, you can see the real benefit of doing this, and that is that we can actually see things that are behind other things after deconvolution. But if you don't do that, then the out of focus light from behind kind of gets in the way of everything, and you can only see the brightest part of your image. So if you want to look at three-dimensional things, you need to use either this confocal or deconvolution microscopy. So in order to do this, how you actually do it, all you need, the equipment you need is a motorized stage. So to take this out of focus image of this bead, we're not sitting there turning the focus knob and taking a picture every second as we turn the knob. We're just not precise enough. So what we have is we have a computer control the position of this stage. So it moves a certain amount, and then it takes a picture, then it moves again and takes another picture. And doing that for the entire maybe 10 microns takes about 10 minutes. But when you're done, we have this very precisely calibrated image of a single bead going in and out of focus. And of course, you need to have very good optics to do this. I'll talk about the actual process later. Okay. I thought what I would do now is show some of the three-dimensional images that we can take with this method. So this isn't the 3D glasses part yet, but this is the source. So this is a similar kind of image where we've moved the stage by a little bit at a time and taken one single image at each section. So each individual section is called a Z section because we're going in the Z or Z direction. And this is 200 nanometers apart. So you can see this is after deconvolution has happened, and so there's not much out of focus light in between contaminating each image. So this is, can anyone tell me, we've seen this before, what are we looking at here? Does anyone know what this is? I've showed you, not this exact image, but something like it before. I haven't shown you too many different things. So this
C. elegans.
Yeah, that's right. Yeah. C. elegans, what is what kind of cells or what kind of chromosomes are these? It's meiosis. Okay, it's the C. elegans meiotic cells, and it's the C. elegans gonad. Yeah. And as we go through in Z, I hope you can see that. So we're going to go all the way to the top of one edge, and you can see all the cells are kind of close to each other. But then as we keep going through, now we only see cells on the sides. And then as we go to the other side, now the cells are close to each other again. So what that means is that this gonad is actually this cylinder. It's a tube of cells that's kind of hollow on the inside. Now we can see a different kind of view of these chromosomes. So once you take this three-dimensional image, of course, if you want to put this in a publication, if you want to show somebody on a piece of paper or on a screen like this, what this looks like, to see the three-dimensional image, I have to step up and down, and you can only look at one section at a time. It's not that easy to see. So a common way of showing this is to do what's called projecting the 3D image. Oh, you need to. Yeah, sure. And there's different ways of doing projection, but one of the most common ways is what's called maximum intensity projection. And this involves, so if you imagine the Z stack going into the plane of this screen, you have, for each section, you have one pixel at each XY coordinate on this screen. And to do a projection of maximum intensity, you simply go down this Z section and keep the maximum intensity pixel. So you keep the brightest pixel and only show that one. So then if there's a bright object here and there's a bright object here, this projection will show both of them on the same plane. So it's pretty conceptually easy to think about. So that's what's going on the left. On the right, there's projections that do that, except they're not taken straight into the plane of the screen. They're just taking a very slight angle. So you take one projection going at a 2-degree angle into the screen, and then you take another projection going at a -2-degree angle into the screen. So because you have this data in a 3-dimensional matrix, you can project along any axis you want. So if you take one projection at this angle and one projection at this angle, and then color them in slightly different colors, one is red and one is blue, cyan, then you can do this trick where each eye sees a different projection. So your left eye is only seeing, I think, the red pixels, and your right eye is only seeing, no, it's the other way around because it's blocking. Anyway, yeah, each eye is seeing only one set of pixels, and that's exactly how stereo vision works. So yeah, so we can see, I hope you can see the 3-dimensional nature of this. Now, it shouldn't matter if you tilt your head or anything like that either, so it's actually pretty robust. I can see it even standing over here. It definitely looks 3-dimensional. So here, by looking, I hope you can see that not only is this gonad a cylinder, but it's actually composed of these rings of nuclei that are stacked one above the other. And yeah, so okay, because we have this 3-dimensional, I should have looped this one. So let me just picture these loop. So that we keep going. Because these are not just single images, but I actually made movies of these. That's the other nice benefit of having. Yeah, it stopped. Well, anyway. Yeah, so I think in the 3-dimensional version of the one on the right, it's pretty obvious that you've got these stacked rings. And that's something that I never really even noticed just going up and down, because when you just go up and down, your brain forgets what you've seen. As soon as you scroll down and lose one Z-section, your brain forgets what that looked like. You can't really grasp the entire 3-dimensional structure of what you're looking at without seeing it all in 3D. Yeah, just trying to convince you that 3D is quite important for studying cell biology, not just chromosomes, of course. And then I have a few more of these that we used. So part of what my lab studies is whether cells can divide correctly or not after they've had DNA damage. And so we have a lot of these cells that are just growing in a dish, and wherever one of the cells has gone through anaphase, we can take a 3-dimensional picture like this. Let's see. These are mouse cells, and so you can see that there's a lot of heterochromatin in these interface cells, and also along the rim of these nuclei, you can also see heterochromatin there. And then in the anaphase cells, I hope you can see in the right chromatin mass there that these cells seem to be dividing. They're not exactly dividing in parallel to the plane, they're kind of a little bit into it. And the centromeres that are heterochromatic, you can see this kind of band of centromeres on the right-hand chromatin mass. And that's where the microtubules are attaching. So yeah, this one is actually, seems to be completely normal. This one, however, there's some problem, right? So this is actually the stage after anaphase. Does anybody remember what that is? What's after anaphase? Yeah. Yeah, so this is a telophase pair of nuclei where all the chromosomes should be back together. The nuclear envelope is reforming. But can anybody hazard a guess? Can anybody guess what's going on here? Why these nuclei look like they're pointing at each other? So, of course, you don't need 3D to see this. You can see it pretty obviously with just 2D. But there's something going on here. Anybody guess what might have happened? If you were a early 20th century cell biologist and you saw this image, could you guess anything about what might have happened here? So clearly the cell divided this way, and most of the chromosomes have made it into the new nucleus, but these two are still stuck together and they're really pointing at each other. So what happened? Those two chromosomes are actually still connected. That's what's happening. So usually during DNA replication, you replicate the entire chromosome, and that means that you can separate the chromosome nicely into two daughter chromatids that each have the identical genetic information. But if something has slowed down replication to where one part of the chromosome hasn't replicated yet, then if you try to pull the chromosomes apart, most of them will go, but where it's not replicated, there will still be, those two chromosomes will still be connected by that unreplicated region. And that's what's going on here. You can't actually see it, but there's very likely just a single strand of DNA still connecting those two chromosomes. And it's strong enough that it's pulling the rest of the chromosome into this this kind of sharply pointed mass, and that's where this one DNA loop, very likely, this one loop of DNA still has not yet replicated. What happens eventually is that DNA doesn't break, because breakage would be really bad. In most cases, that single DNA strand actually unwinds into two single-stranded pieces of DNA, and it gets repaired later. So cells, even though this looks pretty severe, and in fact the cell doesn't really understand as it's happening, and it keeps cell membrane forms in between these two nuclei, and the cells start to move apart. But while that's happening, this DNA can unwind and become single-stranded, and in the next cell division, the cells will probably be all right. So yeah, a couple more of those. And I think this one may be quite a cool one, because yeah, in this case there's another, there's something going on here that you can really only see with this 3D, I think. And that is that there's this bit of chromosome here that's not attached to the rest of the chromatin mass. So there's just, there's another one of these single DNA strands that's trying to separate one chromosome in the middle, but it's not really easily separating the rest of it. So all these chromosomes are fine. These are all coming this way, and all these chromosomes are fine. But there's this little bit in the middle that is still kind of in the middle, and it's not being segregated correctly. So errors in chromosome segregation, some subtle errors like this are really only visible when you look at them in 3D. Yeah, maybe you can see it in 2D too, but it's much more obvious. And then I thought this one was also pretty cool. So it's not DNA, but here you can see what the nuclear lamina looks like in 3 dimensions. So remember, that's this protein shell that coats the inner surface of the nuclear envelope, and a lot of DNA is attached to it. And here you can see that it's actually pretty wrinkly. There's a lot of these kind of cuts and lines in the nuclear lamina. And there's also parts here that you can see these lines here. These are actually holes in the nucleus where a microtubule has punched through the nucleus and made this tunnel that goes, in many cases, it goes all the way from one side of the nucleus to the other side. And that often happens when the nuclear envelope reforms with a microtubule still stuck through the chromosomes. And it's thought that tunnels can sometimes lead to genome instability because it still has nuclear lamina on it, so chromosomes can attach there, but they're not supposed to be attached to the middle of the nucleus, they're supposed to be on the outside. So you see those a lot more in cancerous cells than you see them in wild type normal cells. And then the last thing, the colors are not great on this, but I hope you can see. So this, I've shown you before this ZAG12 protein, which sits at the pairing centers of C. elegans chromosomes, and it moves around a lot in order to promote the search for homology. Yeah, but this is actually a three-dimensional movie that I took. So hopefully you can see some of the three-dimensional character of the C. elegans gonad as these chromosomes are searching for each other. You can see that towards the end of the movie, it kind of deteriorates in quality a lot. Does anybody know why that is? Why it kind of starts out looking good and then it gets really fuzzy and noisy at the end? That's because if you take a movie for a long time on a living organism, you will start to bleach the fluorescent protein. So this is GFP, and the more you look at GFP, the more you are damaging it with fluorescence. And so eventually the signal to noise, the quality of this movie goes down. Especially because I'm taking a three-dimensional movie of this, so at every time point, there's maybe 10 images that we're taking. But anyway, yeah, you can kind of see this. So this is sometimes referred to as four-dimensional imaging because we're taking three-dimensional images over time. Right. So yeah, so this is kind of a summary of why microscopy is good for cell biology. So we can be really specific. We can see any kind of molecule that we want to see. We can see many combinations of molecules, so it's almost always advantageous to look at multiple proteins at the same time. We're almost never interested in just where is one protein, but where is one protein in relation to another protein. And we get the entire three-dimensional picture of a cell. So we can have something like this, where this is really, really huge in specificity because we're not just looking at different protein molecules here, we're actually looking at different modifications of different regions of the same molecule. So histone H3 can be modified by methylation at different parts of the N-terminal tail, and by using antibodies against different parts, we can see, if you can visualize the green versus purple, you can see where each of those type of modified histone is localized, for instance. Okay. So that's kind of a summary of why microscopy is good for chromosome biology. And let's talk a bit about super resolution before. So I'm going to skip super resolution and probably go to a little bit about about computation. So if you compare where microscopes were 100 years ago versus where they are now, or in recent times, when I made this slide, which is already 7 years ago, you can see that a lot of progress has been made, right? It used to be people had to kind of draw what they saw with a camera lucida. And although they learned a lot, and you could arguably say they learned more back then because they were looking at it for the first time, we can really do a lot more now. And it's only been in the past 10 years or so that really that image analysis has really taken off. And so why has it taken so long? Because microscopy itself has not really changed that much. Immunofluorescence was invented back in the 60s. Here, what Mats was talking about, he's one of the inventors of one of the kinds of super resolution microscopy that I work on. Really, the main reason why microscopy has become so powerful in the past decade is not because optics has improved, or techniques have improved, but that computational power has improved. So once the idea was that you had to look at your sample with a microscope. So a microscope was something that you looked into and observed. But when people realized that all you had to do was record the light coming out of your sample with a camera, and then you can do anything you want, process it however you want with a computer, then a lot of new microscopy methods were able to be invented. So the reality is that although I have microscopes in my lab with eyepieces, we spend a lot more time looking at images on a computer than we do looking at images actually on the microscope. So people doing cell biology, or who want to do cell biology, not only have to be able to use microscopes, that's about half of the work, but then the other half is analyzing the images. So image processing and image analysis become really important skills for anybody who wants to use microscopy. And the difference between those two, so those two terms are really often used interchangeably. People say image processing or image analysis, but image processing is usually should be reserved for when you take an image and you enhance it. So you start with one image and end up with a different image that has been improved or enhanced or changed in some way. But you take one image and end up with a different image. Okay, whereas image analysis is slightly different, and that is you take images like the ones I've been showing you. Starting with those images, you do some sort of computation and end up with a bunch of information about what that images contain. And to do quantitative measurements, you really need to do both. But this is what lets you take the huge amount of data present in an image file. So the image files I was showing you can be hundreds of megabytes or several gigabytes each. And if they're movies, then they can be hundreds of gigabytes each for each of these files. But you can't really analyze that. It's too much data for you to analyze. So image analysis, computational image analysis, lets you extract some numbers from that. And so I'll just go over a little bit about how people can do this. And yeah, it's not too early to think about this. I think I've already shown some of this to you. I think I have another class where I talk about some computer stuff, and image analysis is part of that. But I don't think it's complete redundant with that. So there's a really nice image analysis software package that I'd like to recommend if you have any interest in this. It's called, the software is called Fiji, but it's based on this earlier program called ImageJ that was actually written a long time ago by the US government, National Institute of Health, for quantitative image analysis. But whatever computer system you have, you can get this Fiji program. And it has a lot of very, very useful built-in image analysis routines. So for instance, if you have a picture of all these cells and you wanted to measure, for instance, how big or what's the average area occupied by each nucleus, it would take a very long time to do this by hand. So luckily, this program has some really nice built-in ways of doing that. So you can't really see, can't really read this. Anyway, yeah, there are ways that you can just train this software to learn what is a cell nucleus and what is not. Mostly, you have to start manually by just drawing a circle around a few of these nuclei and classifying that as image subset number 1, for instance, or class number 1. And then you also draw a few background circles and call that class number 2. And Fiji, or this part of Fiji, can then learn, okay, everything you circled has a certain intensity and a certain kind of pattern inside it, and that's what I'll call class 1. And everything you didn't or every background bit you circled in green also has a different kind of statistical distribution of intensity. So you don't need to tell it, okay, circle everything that's a certain brightness, or circle everything that has DNA in it. All you have to do is give it a few examples, and it can learn what you mean in a way. You kind of have to do some correction to fill in some holes, but eventually this software can learn, all right, those are, everything in white is DNA, or is a nucleus, and now you can go ahead and analyze further. You can, so it's just really user-friendly, right? You don't have to write any programs to do this. If you want to fill the holes in these binary masks, all you have to do is select this fill holes option, and then you go from this to this, where all these holes have been filled. Then you can do things like find things that are really close to each other and separate them. So every different colored blob now is one nucleus that this program knows about. Right, and then you can finally count how many there are. You can say, okay, this program found 60 nuclei in this picture. And then once you have these regions, you can do things like, what is the area, what is the mean intensity, what kind of standard deviation do you have. So this would be image analysis. This would just be an example of image analysis, where you can take this original image that had a ton of, right, just by looking at this, you can't really conclude anything. But if you take enough of these, then you can get some data that actually has a distribution of intensity. For instance, you might think, okay, these cells are really brighter than all the others, so maybe you'll go and investigate what's going on with these guys. All right, so this is just one example of how to do image analysis. Pretty much for any image analysis routine, you would start by having some method of identifying the objects that you're interested in. In this case, it was nuclei, but in other cases, it might be chromosomes, or it might be spots. It might be spots of RAD51. So the first step is identifying them, and the second step would be measuring them, like measuring their intensity and their shape. And usually the first step is harder, because once you've identified something in the computer, then it's very easy to calculate how bright it is. But the hard part is identifying with enough accuracy so that you don't make any mistakes. And that is actually a little tricky, because our eyes are still a little bit better than computers. For instance, we can look at this and we can say, here's one nucleus here, and here's another nucleus that looks like it's maybe either just starting or just finishing with division. But because those are so close together, the computer ends up making them all into one. And then at the end, there's a single area drawn around this, and it's numbered. So even with this really fancy computer programs that we have, everything is still, you have to supervise and check everything the computer does to make sure that it's not making too many mistakes. In this case, you would just overlay this with your with the original images, and if there was some mistaken one, you can just throw it away or manually correct it or something like that. All right, so that's one way to do it, and Fiji is very powerful, but invariably you will end up having problems that you can't solve with the built-in Fiji software. So eventually writing your own software becomes very important for doing this. Obviously I can't cover that in this class, but for instance, for counting RAD51 foci, I had to write some of my own software that would go from, that would be able to accurately count how many RAD51 foci are in each nucleus. And it does that by first finding these C. elegans nuclei with this interesting methods where the intensity of DNA is kind of concentrated down to a single point and then used to identify the centers of these nuclei. This is kind of way outside the scope here, so I'm just going to jump to the end where I can show you that eventually you get this, you have a bunch of Rad51 spots, and you can associate each group of spots with a single nucleus. So if anyone's interested in more detail about this, I can go over it. But really, those are good ways of analyzing microscopy data. But still, a lot of people, including my lab, has to do things by hand. So this sort of scoring paired versus unpaired chromosomes that I mentioned before, we actually take these gonads in order to get this quantitative data. We actually put some plastic sheet on our computer monitor and circle every nucleus on the computer monitor and put a plus if the signals are paired and a minus if the signals are unpaired. And then, because we can't contain this entire gonad on one image, we then move the plastic sheet and put another plastic sheet there and tape them together. I was going to bring some of those today to show you, but because of the rain, I wasn't able to bring this big plastic sheet, but maybe next time. So both really high-tech and really low-tech methods are both used, and just kind of, you have to decide what is going to take the least amount of time, or what's most efficient in the long run. Okay, so that's kind of my opinions and some of my thoughts on microscopy. And at the very end, I will introduce this completely different way of analyzing the 3D structure of DNA in the nucleus. I think for this, I may need to draw a few things. So there's methods based on DNA sequencing that can also tell you about the three-dimensional structure of the genome. And these have, like most techniques, they have fancy little acronyms. They're known as 3C, 4C, and high C. And 3C, I should just introduce 3C to begin with. 3C stands for chromosome conformation capture. And it's a pretty clever idea that's been around maybe for 10 years now. And it's based on the idea that you can, so inside a nucleus, of course, DNA is sitting on these chromosome loops. So each chromosome has its own chromosome territory. And then another chromosome is sitting here, for instance. And with a microscope, you can look at this, but it all looks like DNA. You can't really tell what, just from looking at DNA, you can't tell which chromosome is which or which sequence is which. But let's kind of zoom up on this region here. So you've got one loop coming in here, and one loop coming here, and another loop coming here. So these are real three DNA molecules in 3D space that are really close to each other. So what people figured out you could do is, instead of looking at these cells with a microscope, what you can do is probe, you can measure the conformation of these DNA strands by artificially linking them together and then sequencing what you get. So there are these molecules called cross-linkers whose function is actually, they can just cause physical links to occur between all molecules, proteins and DNA and whatever. So you kill these cells and then you add this chemical cross-linker. So all the DNA is kind of locked together wherever it is. So you can imagine all this DNA is sort of locked in place with these cross-linked molecules. The next step is to use a restriction enzyme, which I've introduced before. It's just an enzyme from bacteria that cuts DNA in a very specific sequence site. So wherever this particular sequence happens to be in the DNA is where it gets cut. So, so this DNA that's been locked together now gets cut with this restriction enzyme. And if you remember, the restriction enzyme sequences are usually palindromic. So if the sequence that it cuts were this sequence, for instance, then it would result in one molecule with this overhang, so this GC overhang, and this part of the molecule is free to rebind to this part of the molecule, or it can rebind to another DNA molecule nearby. So the next step is to rejoin these cut DNA strands. But the cool thing is that they don't necessarily rejoin back to where they started. They can rejoin, let's say there's been another cut here. This DNA molecule, they're still kind of flapping around in 3D space. So this DNA molecule and this DNA molecule might join with each other instead after putting these molecules together. So what you've done now is you've created a link between two DNA molecules that were close to each other in space. Okay, this molecule and this molecule, or this one and this one, they happen to be close by each other in space, but they're not on the same chromosome, or they're not on the same loop, but they were close to each other, and now they're physically linked together. Okay. Now what you can do after you've done this, if you, for instance, if you wondered, does this DNA molecule ever localize close to this one? Now you can do PCR with this, with a primer here and a primer here, and if you get a PCR product based on these two DNA sequences, then you'll know, okay, in this nucleus, this piece of DNA and this piece of DNA were close to each other. And so this is the first level of 3C, or chromosome conformation capture, where you are probing, okay, do these two DNA sequences ever come close to each other? And you can find out if they do by joining them together after cutting this restriction enzyme. The next level, 4C, is a little more complicated, so I'm not going to talk too much about it. But instead of just using a PCR product, so all these techniques have this. They start the same way with cross-linking and cutting and rejoining the DNA in the nucleus. The rest of the technique only differs by how you detect the output. So 3C is very useful if you're just asking, does DNA sequence A ever come close to DNA sequence B? And depending on how often they're religated together, you will get the amount of PCR products will change. But once high throughput DNA sequencing became cheap enough to consider this, so sequencing the entire genome of cells, people realized instead of using PCR to detect this sequence, let's just sequence all of this cross-linked DNA and see what has become close to what. So we already knew, so if we have the entire genome sequenced, we already know this gene, we already know this gene. They're not supposed to come together, they're not supposed to be on the same chromosome. But if your sequence, if in the full genome sequence, you get gene B and then immediately after you get gene A, when that's not how they actually are on the chromosome, then you know that this, and also you know the site of the restriction digest, so you know that there's a GGCC right here. So when you pick up these sorts of molecules in your full genome sequence, then you know that this chromosome or this bit of DNA and this bit of DNA were close to each other in the nucleus. So this method, just by forcing these DNA molecules that are somewhat close to each other in space, by connecting them together, that's essentially what this technique does. you are connecting and making a single unbroken DNA molecule from DNA molecules that were just beforehand, they were just close to each other in space. When you have all of that, then you can analyze the 3D structure of a chromosome or the entire nucleus without actually looking at it. And because it's at the sequence level, it's much higher resolution than you can get with a microscope. The problem is you can't really get three-dimensional XYZ data out of this. The kind of data you really get is this sort of interaction data where here's one chromosome, for instance, and you can see how often is this gene, for instance, how often is this gene touching this other gene on the same part of the chromosome. Here, where it's red, or here, this part, this darker red part, is where there's a high level of interaction. So this is just on one single chromosome, and you might expect, of course, one chromosome is folded up in this sort of loop structure, that wherever these loops are touching, you would get some sort of high level interaction like that. But you can actually do this on the entire genome as well. And this is what that looks like. So this is an example high-seq output for the entire human genome. And so what you see here, each row is one of the 23 chromosomes, and each row in each column is also one of the 23 chromosomes, so it's symmetrical. You can see that each chromosome interacts with itself much more strongly than it interacts with the rest of the genome. So wherever it's really dark, this is, say, chromosome 16 interacting with chromosome 16. So you get a lot of sequence where one bit of chromosome 16 has ligated, connected to another bit of chromosome 16. But there's also a lot of other little bits. So this chromosome here, although it tends to interact with itself most, there are some parts of it that like to sort of probe and make connections with other chromosomes. So actually, by analyzing this sorts of images, or this sort of connection data, people have come up with not actually a picture of what the genome looks like, but a kind of model. It's not a really high resolution image of what the genome looks like, but by examining, for instance, the degree of interconnection between chromosomes, people have learned how much, for instance, how much surfaces between chromosomes should be touching to each other. Okay, so that's kind of, yeah. So people are working on, okay, I guess the main limitation of this, other than the fact that it's not an image, is that it's very difficult to do this on one cell at a time, because as I showed you, you break DNA and then you reconnect it randomly. But of course, there's a chance that this DNA will just reconnect back exactly where it came from, and then you won't get any information about conformation from that. You only get new information when this DNA ligates with a different DNA partner. And if you are looking at a single cell, well, for each DNA piece, you only get one chance to get that information or not. So still the idea of intricately mapping the entire genome in a single cell is still kind of out of reach with this method. But who knows, it's improving very rapidly. So there may be some ways of actually getting information from every single piece of DNA inside the nucleus. But for now, we just get some complementary information to microscopy. Okay, I think that will do for today. It's actually, that's the end of the show. So, yeah, if there's any questions, you can ask me now or after this. And yeah, next week there will be a little bit about chromosomes and cancer, but then most of it will be a review over this entire class, this entire course. And then there will be a test on the 25th. I have also quizzes from last week. Which everybody did well on, so. you can keep these because I have many, so you can take it home.
Thank you.
Yeah. I still have to
Oh.
get back to you.
If I take your next semester lecture, practical computing for
yes.
biologists, will I became will I learn to be good at computing? Very difficult. You said write your own programs.
It's very introductory.
Introductory.
Yeah, you can ask Anna about it because she had that class before. It doesn't get that complicated, but I'm happy to talk about more complicated things if it's too easy for you.
Thank you.
Yeah. It mainly focuses on Unix and then some Python, but it's really kind of introductory to some bioinformatics.